Experiment 1. Use of insects as substitute of fish meal.
The idea behind this experiment is to provide them as food to the fishes as an alternative to fish meal.
This is the new concept in the field of aquaculture, as the insects contain very high amount of proteins and lipids and require minimum input.
3 insect species were used in this experiment. First insect, Grylus.
Grylus is common in France and has an Asiatic origin. It grows to a size of 2 to 3 centimeters and requires a temperature of 25 degrees Celsius.
A substrate of leaves, apples, and vegetable peels is made for the insect to feed and lay eggs. The larvae hence produced are used in the fish feed.
Grylus is preferred because it has 62% of protein, 44% of lipid, and 70% of water. It has a good FCR. For every 10 kg of food, we get 9.9 kg of Grylus.
The second species, Tenebrio. It is widely distributed in Europe. It attains a size of 1.5 centimeters. It reproduces quickly in about 2 to 3 months at 25 degrees Celsius.
Insect undergoes metamorphosis, forming nymph. The larva is vermiform and yellowish white in color. The juvenile is brownish orange and the adult is black.
They are kept inside the egg trays. Tenebrios feed on corn flour and hence they are also known as flour worms. They contain 50% of protein, 35.8% of lipid and about 62% water.
The third insect, Hermatia. Hermatia has an American origin and the adults attain a size of 1.5 to 2 centimeters. They feed on vegetable peels and animal waste, commonly known as black soldier fly.
The adults prefer humid conditions to lay eggs, and as the larvae metamorphose, they try to avoid this condition by escaping. These larvae are collected and then transferred to another box, from where they can be used in feed.
This new technique can be adopted in India by the farmers to enhance their production, as the insect larvae contains all the essential nutrients which are beneficial for the fishes.
The second experiment was cultivation of different plants by aquaponics using tilapia as the fish species. The fruits and legumes cultivated were barley, basil, lettuce, and strawberry. This experiment includes utilization of nitrogenous waste as well as organic matter from tilapia to act as basis of plant cultivation. For their growth, the plants require nitrates and phosphates, which are provided to them from fish excreta. The organic matter obtained from fishes act as manure, hence avoiding the need of fertilizers. In return, the plants reduces the concentration of toxic waste from water, thereby completing the biological cycle.
Tilapia of 300 to 400 grams of body weight is cultured in ratio of 2 male is to 6 female for 1 cubic meter area. The spawning occurs naturally in every 15 days at 26 degrees Celsius temperature and has an incubation period of 4 days. The waste excreted from fish by providing 30 to 40 grams of feed helps in producing plants in 1 meter cube area.
The idea behind this experiment is to determine the amount of nitrogenous waste used from tilapia to grow legumes and fruits in per meter square area.
Experiment 3. Using worm filter to remove organic matter from sturgeon tanks to get filtered water.
This is the recirculatory system to produce sturgeons containing 6 tanks. The water in the tanks is received from the river. The series of filters is installed in the recirculatory system along with the worm filter to remove the organic load from the water.
The water from the tank goes to the Hydrotech filter, which has an efficiency to remove 50% of organic matter. The remaining 50% of water is sent to the UV filter, sterilizing the water. Then from here, it goes to the biological filter, further cleaning the water before supplying it back in the recirculatory.
The 50% of organic load left in the Hydrotech filter is stored in a sump and sprayed into the worm filter 2 to 3 times a day. This worm filter consists of earthworms which breaks down the complex organic matter, forming simpler compounds in water. The water is then passed through a bacterial bed, reducing the bacteria and hence purifying the water completely. This water is used for growing legumes and strawberries in the greenhouse, hence providing nutrient-rich water to the plants.
The motive behind the working of the recirculatory system with the worm filter is to fulfill two purposes. Firstly, to remove the organic load from the sturgeon tank, providing them clean, filtered water, and secondly, to provide nutrient-rich water to the plants in the greenhouse for their growth and nourishment.
On being attached to Apiva, we gained a lot of knowledge of aquaponics, working of recirculatory system, and about water purification.
And we would like to show a heartfelt gratitude to Dr. Catherine for giving us this privilege and guiding us throughout. We would also like to thank Jerome and Quanto for the support and help.
Messi.